tubes, or bottles prepared from each lot or batch should be incubated for 2 to 5 days at 35 to 30 degrees centigrade and 50 to 55 degrees centigrade. For a lot of 100 or less units, 3 to 5% units should be tested. For a larger lot, 10 random plates or tubes should be tested for sterility. If there is no microbial growth after incubation, that batch or lot can be considered sterile. Discard all sterility samples when the tests have been completed. Growth performance. To test whether the medium prepared supports the growth of bacteria, inoculate a few units with appropriate stock cultures or fresh isolates. Use a standard inoculation procedure. For testing new lots or batches of media, inoculate old and new lots together and compare the performance of the two lots side by side. Stability. The earlier mentioned procedures should also be performed on stored prepared media in order to determine whether the storage conditions are optimal or not. If a medium does not give satisfactory results, even after following all the manufacturer's recommendations, then these steps should be taken. Record the nature of the problem and the method of preparation of the medium. Note the lot or batch number and the date it was received. Call the technical support number of the supplier. Preparation of basal media. Peptone water. This medium is chiefly used as the basal medium for carbohydrate fermentation tests. It is also used to test the formation of indole. This is usually supplied as a golden granular powder with a low moisture content and gives a pH between 5 and 7 in a 1% solution. It is hygroscopic and hence the stock bottles should be kept firmly closed. Follow the manufacturer's instructions for the reconstitution of the medium and all the directions for preparation, sterilization, and storage of dehydrated media as mentioned earlier need to be adhered to. Alternatively, if you are preparing your own Peptone water, then the recipe for the medium is Peptone 10 grams, sodium chloride 5 grams, and water 1 liter. Dissolve the ingredients in warm water and adjust the pH to 7.4 to 7.5. Filter and distribute as required. Autoclave at 121 degrees centigrade for 15 minutes. Nutrient agar. Nutrient agar is solidified nutrient broth. Reconstitute the dehydrated nutrient broth according to manufacturer's instructions. Add 2% agar to the solution. Heat to completely dissolve the agar without burning it. All the directions for preparation, sterilization, and storage of dehydrated media, as mentioned earlier, should be adhered to. Preparation of enriched media. Blood agar. Blood agar is widely used in medical bacteriology. Apart from being an enriched medium, it is also used as an indicator medium to study the hemolysis of bacteria like Staphylococcus aureus and Streptococcus pyogenes. It is generally poured as plates. Horse blood or sheep blood is most commonly used. Human blood is not recommended. Blood used for the preparation of blood agar should be as fresh as possible and should have been stored at 2 to 8 degrees centigrade, taking care that it is not frozen. Defibrinated blood is recommended for use rather than blood containing an anticoagulant. Concentrations can vary from 5 to 50%, although 10% is the most commonly used concentration. Prepare and sterilize the nutrient agar base as mentioned earlier. Warm the blood in a 35 degrees centigrade incubator before addition to the nutrient agar base, which has been cooled to 50 degrees centigrade. Add 10% or 100 milliliters blood to 1 liter of sterile agar base. Agar will tend to settle to the bottom of the flask during sterilization, so all flasks should be swirled in large circle to facilitate proper mixing of the agar. Adequate mixing in a large vessel is also essential to ensure proper aeration of the blood. Properly aerated blood agar is cherry red. After a homogeneous mixture is obtained, get ready for pouring into plates. Follow the precautions mentioned in the distribution of media earlier in this section. Any bubbles on the surface of the agar should be removed by passing the flame of the Bunsen burner across the surface of the medium. After pouring the media into plates, leave them undisturbed till agar is solidified. Then, stack the plates and seal them in plastic bags and store at 4 degrees centigrade. Chocolate agar. This medium is prepared from blood agar by heating and contains certain extra nutrients required for the growth of fastidious organisms like Neisseria and Pneumococcus. The heat causes red cells to lyse and release the nutrients. Prepare and sterilize desired amount of nutrient agar. Cool it in a water bath at 75 degrees centigrade. Add 10% sterile blood and allow the medium to remain at 75 degrees centigrade, mixing the blood and agar gently from time to time till the color of the medium turns chocolate brown within about 10 minutes. After a homogeneous mixture is obtained, get ready for pouring into plates. Follow the precautions mentioned in the distribution of media earlier in this section. Any bubbles on the surface of the agar should be removed by passing the flame of the Bunsen burner across the surface of the medium. After pouring the media into plates, leave them undisturbed till agar is solidified. Then stack the plates and seal them in plastic bags and store at 4 degrees centigrade. Preparation of indicator medium McConkey agar. This is a useful medium for the cultivation of Enterobacteria. It contains bile salts which inhibit the growth of gram-positive bacteria and promotes the growth of only gram-negative bacteria. In addition, it contains a dye called neutral red, which gives a pink color to colonies of lactose fermenting bacteria. Non-lactose fermenting bacteria, on the other hand, form colorless colonies. Dehydrated McConkey medium is available in the market. It should be reconstituted according to the manufacturer's instructions. All the directions for preparation, sterilization, and storage of dehydrated media, as mentioned earlier in this section, should be strictly adhered to. However, if you choose to make your own medium, the recipe is as follows: peptone 20 grams, sodium taurocolate 5 grams, water 1 liter, neutral red solution 2% in 50% ethanol 3.5 milliliters, lactose 10% aqueous solution 100 milliliters. Dissolve the peptone and taurocolate in water by heating. Add the agar and dissolve it in an autoclave. Adjust the pH to 7.5. Add the lactose and neutral red. Shake well. Heat in autoclave with free steam at 100 degrees centigrade for 1 hour and then at 115 degrees centigrade for 15 minutes. Pour plates as described earlier. The medium when set is light pink in color.